Increased mtDNA mutations with aging promotes amyloid accumulation and brain atrophy in the APP-LD transgenic mouse model of Alzheimer's disease, which basically means that mitochondrial DNA mutations promotes amyloid plaques that form within the brain and then causes atrophy, which is similar to Alzheimer's disease. Alzheimer's disease is a progressive neurodegenerative disorder that is most commonly cause of dementia in the elderly. A major feature of AD is the presence of amyloid plaques, which are basically proteins that are sticking together, and this is caused by an increase in the peptide beta amyloid. The beta amyloids consist of 42 amino acids and are derived from a larger molecule called the beta precursor protein, or APP. There are two enzymes that are essential for cleaving APP and producing the AB42. There are BACE1 and gamma secretase. BACE1 will cleave APP, which is found in the cell membrane outside of the cell, reducing a peptide called C99, and the gamma secretase will cleave inside of the cell, and then the resulting peptide is the AB42, which will then create the amyloid plaques. AD patients seem to have these amyloid plaques and are causing neurodegeneration, but the mechanism behind the plaques and neurodegeneration is unknown. AD patients also have higher levels of mitochondrial DNA deletions in neurons of the hippocampus and neocortex with age, and in vitro studies have provided evidence that AB toxicity may cause changes to morphological, chemical, and genetic changes to the mitochondria itself and the mitochondrial DNA. The goal of this study is to determine how beta amyloids and their toxicity as they create these plaques are actually interacting with the mitochondrial DNA, and also using that idea of age and in association to the mitochondrial DNA dysfunction. So they're going to use knock-in mice that have a mutation that inactivates the proofreading function of mitochondrial DNA, which has been shown in previous studies. The poly-G-A is essential for preventing mutations with age, and so the poly-G-A mice are going to show premature aging since they have a mutation within that polymerase. They're going to cross the mice with the poly-G-A mice with transgenic mice that overexpress APP due to the London familial AD mutation. And these APP-LD mice do not exhibit neuron loss but do develop amyloid plaques at 1 year. And so the interaction between the APP-LD mice and the poly-G-A mice are going to be able to show how aging and dysfunction of the mitochondria is interacting or affecting the AB toxicity and the processing of the APP. They're going to use 4 genotypes. They're going to use homozygous D257A, which has the homozygous poly-G-A mutation, homozygous D257A, which is also transgenic for APP-LD, and the APP-LD transgenic mice, and then a wild type. Once the mice were crossed at 12 months old, the brains were analyzed with ELISA, which is an enzyme-linked immunosorbent assay, which uses antibodies and color changes to identify substance. So in this case, they were identifying the presence of AB42 peptides. And they found that in figure A, when they were looking at the analysis, that the D257A APP-LD had a twofold increase in the AB42 peptides. So they wanted to see if that increase was due to the APP gene being overexpressed due to the mutation of the poly-G-A. And so they did a Western blot analysis of the whole brain, looking for full-length APP, and they found that actually, which you can see in figure C, those that had the mutation for the D257A and APP-LD transgenic mice actually had 40% less full-length APP. So there was some sort of synergism that was taking place between the mutation and APP that was actually reducing and altering those levels. They're not sure why that occurred, but further research would need to occur in order to understand if there was actually an influence in the transcription or the translation of the APP protein. To determine if the twofold increase in the AB42 level caused by the mitochondrial mutation led to an increase in amyloid plaque number in the brain, the brains were sectioned from the same 12 mice, stained, and then counted for the AB42 positive plaques. They found small and sparsely distributed plaques throughout the cortex and the hippocampus, which were expected in the D257A APP-LD mice and also in the APP-LD mice, which is shown in figure A. But they also found plaques in the thalamus of the D257A mutant mice, and those mice also had much larger and denser plaques in comparison, which you could see in figure A here, those that have the mitochondrial mutation. So overall, this shows that AB42 levels in amyloid plaque load were increased in the brain when there was a mitochondrial mutation and an increased level of APP for the mice. So therefore, the D257A mutation worsens the cerebral accumulation of AB42. This increase in amyloid load could have been caused by an increase in the AB42 production or a decrease in the AB42 clearance. So to measure the AB42 production, they did this by an analysis with the Western blot, looking at the AB42 producing enzymes, BACE1 and the gamma secretase. They found that for the cells, which you can see in B here, or the mice that had the mitochondrial mutation, had a slight increase in the BACE1, and though compared to the wild type, but it was not significant. And then in H it shows that those for the gamma secretase, again another enzyme that produces the AB42, there was no significant increase in those enzymes compared to the wild type. They also wanted to see if the BACE1 was just more active in comparison to the wild type. So they looked at the C99-C83 ratios, because C99 is the peptide created after the BACE1 cleaves APP. A higher ratio would have indicated that the BACE1 has increased activity, which is not observed, which is shown in D through E down here. So in conclusion, there's no changes in the enzyme AB42 production, or the enzymes that produce this, suggesting that the AB42 level increase in plaque load was unlikely a result of an increase in the AB42 production. To determine if the AB42 increase was due to a decrease in clearing enzymes, they next looked at IDE and NEP, which are the two major AB42 clearing enzymes in the brain. They did a Western blot for both of them, and they found that the IDE was elevated in response to an increase in AB42 in the APP-LD mice, which has been previously shown as a feedback mechanism, which you could see here, this increase for the APP. But they found with the mice that had the D257A mutation in the mitochondria here and here actually had a decrease in the IDE enzyme. But there was no change in the NEP enzyme or clearing enzyme based off of the different genotypes. So this suggests that there's some sort of inhibition of the IDE, which should be increasing when there's an increase of APP. And that is what's causing the increase in the AB42 levels. Since AD patients do experience atrophy, they stained the brain sections and found that there was a 20 to 30% shrinkage of the brain that occurred only in the D257A APP LD mice, which is shown in figure ABC of figure 5. This suggests that there is a synergism between the effect of the D257A mutation and the APP-LD transgene expression, because they did not find that shrinkage with just the mutation alone or the increases in APP. They also found in figure 6 that there were morphological features that pointed to severe state of degradation of the neurons. They found that the clear cytoplasm swollen nuclei suggested apoptosis was taking place within the cells. To confirm apoptosis and severe degradation of the neurons, caspase activation was measured, which is a biochemical marker for apoptosis. A Western blot was used to show that the D257A APP-LD was 50% higher reading of capsase in those cells, which would result in an increase in apoptosis and could be part of the reason for the severe atrophy that was taking place. P25 is also associated with neurodegeneration and apoptosis in humans and animals. So Western blot was used to measure P25, and they found that it was 50% higher in the brains of the D257A APP-LD mice compared to the wild type, again suggesting a reason for atrophy and confirming the neurodegeneration taking place in the D257A APP-LD mice. Since the D257A mice had considerable atrophy, they checked for neuron loss through neuron counting. They found that the 4 genotypes did not exhibit significant differences in the neuron density, which follows previous research which has shown that the APP transgenic mice typically do not experience neural death, but will show signs of neurodegeneration such as synaptic loss, Cas space activation, and increased P25. And this might just be because they have the toxicity of the amyloid plaques that form at 1 year, and so they really just don't have the onset of neuron loss because they usually die at an earlier age. Overall, the study has found that the AB42 levels in the amyloid plaque load increase in the brains of the D257A APP LD mice, and the D257A mutation prevented the AB-induced IDE increase. So there was no way to clear the AB42 peptides that were building up, which may be responsible for the worsening of the amyloid accumulation. Overall, this study points to the idea that AB42, or the amyloid buildup alone, is not substantial, or is not a substantial cause for the neurodegeneration alone, but it's the two factors of the amyloid buildup and the mitochondrial dysfunction combined together that actually leads to the neurodegeneration. So this could be extremely important for potential treatments to reduce or to avoid and prevent neurodegeneration in AD patients. Further research would be needed to confirm the actual cause for the decrease in the APP-LD production and the D257A AP-LD mice to see if there was any sort of link between the transcription and translation of the full-length APP and why it was reduced 40% in those mice. It would also be essential to determine if AB could actually worsen any pre-existing age-related mitochondrial mutations or defects and deficits, and how that would affect neurodegeneration. And then finally, to get a clear sequence of the progression of neurodegeneration in the D257A mutation APP LD transgenic mice to see how atrophy and apoptosis actually progresses through the lifespan would be essential to get a clearer picture of this synergism and interaction between mitochondrial dysfunction and the amyloid buildup.